Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we will interview one or more authors from the feature articles, the papers of the month, or other articles published in that month's issue of the Diabetes Journal. We also have a Sweet Talk segment of the podcast, which is our open format portion of the podcast that can include topical interviews or discussions. My name is Darlene Sandoval, and I'm joined by my podcast co-host, Dr. Kirk Habiger and Dr. Kevin Williams. How are y'all doing today?
Hey, Dar.
Hello.
Kevin.
Hello.
So this month, we're recording in March, which is Women's History Month. So besides me, of course, I would love to know who is your most inspirational.
Dang it, you took my choice.
Who is your most inspirational woman scientist, live or dead? Kirk, we'll start with you.
Yeah, I've got, I think I've mentioned this before, but I have a house full of teen and preteen boys. So I specifically chose Hedy Lamarr. And she was an Austrian-American actress, but more importantly, she's credited with inventing frequency hopping, which is the technology that we use for Bluetooth, Wi-Fi, and lots of other cool electronic devices that my house could not evidently exist without. So, Hedy Lamarr.
So taking a very personal note there for your selection.
Pragmatic.
Yeah.
Nice.
Kevin, do you have one?
Yeah, I'm going to cheat a little bit. I recommend Rosalind Yallow. We talked about her last month on the podcast, and I think that was a really interesting discussion about her and the development of radioimmunoassay, her background in physics, not in biology, but linking up with Saul Burson on the medical side in order to investigate insulin biology and the development of the radioimmunoassay. And so I think there was a lot of foundational research that we should appreciate and go back and look. And we take for granted today too much about what we know about insulin biology and how little was known, and how great this technique has advanced the field of insulin biology and its understanding, but also contributed to other aspects of physiology. So I would really encourage our listeners to go check out that episode.
Yeah, including GLP-1 physiology, right? Without the insulin immunoassay, we wouldn't have stumbled upon, hey, there's this gut factor that stimulates insulin secretion.
Right.
Yeah.
Right.
Yeah, so on that note, I'm actually going to choose two. Well, I'm going to cheat and choose three. I have to add the third one.
Wait.
Yeah.
Fine, I guess because it's International Women's Month.
Yeah, that's right.
Yeah.
So Svetlana Majithia, the GLP-1.
Yeah.
Right.
Great choice.
And then also Lada Knudsen for her work on GLP-1 as well. Like I really like to highlight their contribution. Not to discount Dan Drucker and Jens Holtz, right? And Stephen Bloom and Randy Seeley, et cetera. But those are two women that were really pretty critical in pushing the physiology forward for Svetlana and pushing the pharmacology forward for Lada.
Absolutely.
I gotta give a shout out to my gal Barbara Corky. I've had the opportunity to interact with her a lot over the last few years, and she has a really interesting story. So I don't know if you all knew this, but she worked as a technician in the lab for years before she went to graduate school. So she was, I think, in her late 30s, early 40s when she went to graduate school. So it's pretty cool. And then to start her major career so late, and then to still have such a huge impact on the field, right?
True.
Yeah, she's been a mentor to so many scientists through her lab, but then also through societies and out in general. And it's really great to see her still active and contributing, giving back.
Yeah, for sure. All right. So what do we have in store for the podcast today? Kirk, what are we doing for our interviews?
Yeah, so today we're going to discuss one of our feature papers from the April issue. We're going to be joined by corresponding author, Dr. Majithia, who's an associate professor in the Department of Medicine and School of Medicine at UCSD. He'll be discussing his paper, An Alternatively Translated Isoform of PPARG Gamma Proposes AF1 Domain Inhibition as an Insulin Sensitization Target.
I have to say that interview actually, I went reading that paper, I almost didn't understand a thing, and that interview was really fun.
Oh, he was great. He was great. Very, very good scientific communicator, right?
Yeah, for sure.
So that's a good one. We're also going to chat with lead author Lahari Basu, who's a PhD candidate, and her mentor, Dr. Jennifer Bruin, associate professor in the Department of Biology, Institute of Biochemistry at Carleton University. And their paper is titled Cisplatin Exposure Dysregulates Insulin Secretion in Male and Female Mice. So a couple of good articles this month as well.
Sounds great. Kevin, we have a very special Sweet Talk episode this month, so tell us about it.
Yes, today we're going to celebrate the life and legacy of Dr. David Wasserman from Vanderbilt University. He had a real lasting impact on the fields of the study of exercise physiology, glucose metabolism, and diabetes. Sadly, we lost him in June of last year, but his impact continues to resonate in the field. We're really going to try and celebrate him in his science, but also him as an individual. And joining us for this discussion are Drs. Maren Laughlin from the NIH, Dr. Julio Ayala from Vanderbilt University, and Dr. Shawn Burgess from UT Southwestern Medical Center. I think this will be a great discussion, so be sure to stick around and find out more.
All right, let's dive in. Next up, we're going to discuss another paper published in April's print edition of Diabetes titled Cisplatin Exposure Dysregulates Insulin Secretion in Male and Female Mice. We are joined by corresponding author Dr. Jennifer Bruin, who is associate professor in the Department of Biology and Institute of Biochemistry at Carleton University, and lead author and PhD candidate Lahari Basu. And for this discussion, Dr. Kevin Williams is going to step in for Dr. Habiger, who's on spring break and hopefully enjoying his time on a beach somewhere with his family. Thank you all for joining us today.
Thank you for having us.
Hi, Dr. Bruin and Lahari. Before we dive into the science, we always like to start off with a little bit of an icebreaker question. And so the question I have for you is that if you weren't in science, what career do you think you might have pursued instead? Let's start with Dr. Bruin.
I would definitely own a bookstore.
Nice.
Nice.
Yeah.
I think in another life, I would be an architect or a first grade teacher.
Oh, wow. That's very diverse.
Yeah.
I'm keeping my options
Or
open.
distinct. Yeah. Awesome. All right. Well, let's jump into the science. So, Dr. Brin, before we discuss the current article, we like to give our authors the opportunity to highlight any other contributing authors or collaborators on this work.
Yeah, thanks for the opportunity to do that. This paper, as you can see, has 19 co-authors, which is the most co-authors for any paper coming out of my lab so far. So this was definitely a collaborative effort. A couple of people I want to highlight. One is an undergraduate student named Kelsey Mackay. She was the student who started this whole project, and this project, which has become a main research stream in my lab. So Kelsey started in my lab in 2019, and she was the one who came up with the idea that we should test the effects of cisplatin on islets. The lab next door to mine is a cancer biology lab, so this seemed like a great little honors thesis project that turned out to be amazing. And every time we do an experiment with cisplatin, we're blown away by the phenotypes. So this has turned into a big research stream that's funded by Diabetes Canada and now has 2 full-time PhD students working on the project. And
Wow.
So Lahari, who obviously you're going to hear from in the podcast, is the lead author on this paper. But I also want to highlight the second author, who's the other PhD student working on this, and her name is Lily Greco-St. Pierre, and she contributed all the human islet data. And I also want to mention that there are 9 co-authors from the lab on this study, just showing how everybody really came together to help make this project happen over many years. And also several labs that we collaborated with who helped us fill in gaps for techniques that we don't have up and running in the lab. So Jan Menningen's lab helped us with Seahorse. Pat McDonald's lab hosted Lahari actually for 8 weeks at the University of Alberta, and she learned how to do electrophysiology in his lab. And then Carol Yock and John Stead here in Ottawa helped us with the transcriptomics.
Nice. An example of how science is a team sport.
Exactly.
And my ears perked up for electrophysiology.
Yeah.
Near and dear to my heart.
All right. So, Dr. Bruin, why don't you give us an overview of, I mean, you touched on it a little bit, at least, of where your lab's going next, but give us an overview of your research program and how this project fit into that.
Sure. Yeah, so I would say most broadly, my lab is interested in the pathogenesis of diabetes and why diabetes rates are continuing to increase globally. So I have a unique background. My undergraduate degree and my PhD were both in toxicology. But for my postdoctoral fellowship, I wanted to really switch gears and learn more about diabetes. And so for my postdoc, I went to UBC and I worked with Tim Kiefer, and we were trying to differentiate stem cells into beta-like cells for treating patients with type 1 diabetes. So nothing at all that I'm currently doing. But at UBC, I really learned about all of the diabetes models and islet biology and how beta cells develop. And so when I started my lab at Carleton in 2016, that's where I decided to, bring all of these different aspects of my training together to develop a really unique niche. So we're interested in how exposure to toxicants impact islet health and subsequently diabetes risk. So the bulk of my lab studies environmental pollutants. We're mainly focused on the most persistent pollutants, the ones that really stick around in the environment, like dioxins and PFAS, which are sometimes called forever chemicals. And so we're looking at how environmental pollutants impact, dysregulate insulin secretion and contribute to diabetes risk. And then the other half of my lab, run by right now 2 PhD students, is looking at the impact of chemotherapeutics on islet health and diabetes risk.
And Lahari, building on that, in your article, you lay out a really compelling link between these chemotherapeutic agents, particularly cisplatin, and diabetes risk. For our listeners, can you walk us through what first motivated you to study these effects of chemotherapeutic agents, particularly cisplatin, on pancreatic islets? Was there something specific in cancer survivor data that pointed you in that direction?
Yeah, absolutely. So there is an increasing body of evidence that shows that cancer survivors have an increased risk of developing type 2 diabetes. And this is irrespective of their age, the type of treatment they received, or even the type of cancer that they had. But there aren't a lot of studies really investigating how exactly this relationship has come to be. And so we started on this hypothesis that potentially it's the treatment or the chemotherapy that the survivors are receiving that's contributing to this increased risk of type 2 diabetes. There are various off-target effects of chemotherapy that have been documented, but there really hasn't been a lot of studies looking at the effects of chemotherapy on metabolic function specifically. And we chose cisplatin because there were quite a few studies that showed that individuals that received cisplatin actually had an increased rate of developing metabolic syndrome compared to individuals with the same type of cancer that received alternate treatments, such as surgery or radiotherapy. We also chose cisplatin because it's one of the more common chemotherapeutic agents. It's used around the world and it treats a wide variety of cancers. So we wanted to start off with something that was a bit more broad to really dive into this relationship between chemotherapy and metabolic function.
Nice. Let's jump into the methods now. So tell us a little bit about the methods for this particular study. I think one of the things that I always love to see that you did very nicely here is a combination of the in vivo and in vitro methods, or ex vivo methods. So tell us a little bit about what you did to explore your question of interest or your hypothesis.
Yeah, so I think the use of the 2 models really helps us approach this question of how exactly is cisplatin affecting islet function in 2 different perspectives. With the in vivo model, we understand how cisplatin is affecting metabolic function from a whole body context. But when we saw that profound effect of cisplatin on plasma insulin levels by isolating the islets and exposing the islets to cisplatin in vitro, we're able to better understand more direct effects that cisplatin is having on the islet itself without sort of the other confounding physiological factors of an in vivo model. And also by isolating the islets and treating them directly with cisplatin, we're also able to uncover and answer more questions related to the mechanism by which cisplatin is affecting islet function.
You know, one thing I found particularly interesting was that cisplatin exposure appeared to disrupt insulin secretion, but the glucose tolerance remained unchanged. Can you walk us through that result, what it might suggest about potential compensation of other tissues?
Yeah, this is definitely a result that also makes us think too. And we have a few hypotheses about why this might be occurring. The first is that at that particular time point, it was very early after exposure. So our first hypothesis is that maybe cisplatin is having a profound impact on plasma insulin, but maybe that hadn't manifested in changes in glucose tolerance yet. We also thought about potentially cisplatin as having an impact on other endocrine hormones such as glucagon. And Kelsey, actually in an earlier cohort, she had measured fasting glucagon about 8 weeks post-exposure in cisplatin-exposed mice and found that cisplatin-exposed mice actually had increased fasting glucagon, which doesn't necessarily help us explain the lack of hyperglycemia that we would expect to see with both increased fasting glucagon as well as decreased plasma insulin. But one thing that we haven't looked at yet is glucagon sensitivity. And so we have another working hypothesis that perhaps there's changes in glucagon sensitivity that is contributing to the lack of change in glucose tolerance despite the change in plasma insulin.
Very cool. And actually, just as an aside, we're interested in glucagon sensitivity as well in terms of high-fat diet. So maybe we should talk offline.
For sure.
Have some fun experimental designs. But you also observed in vitro. So now let's go from the glucose tolerance to these in vitro studies. And you show some very nice, and it was repeated in a couple of different models, but this dysregulated insulin response that you mentioned, and also reduced oxygen consumption in the mouse islets. And what was cool is that, at least with the impaired insulin secretion, this was replicated in the human islets, right? So talk a little bit about these findings, what exactly you saw and what the significance might mean for beta cell function, especially kind of linking that impaired oxygen consumption to the insulin secretion data.
Yeah, for sure. Well, I think one of the key pieces of data that really helps us understand how cisplatin is affecting islet function, or rather how it's not, is that we didn't see a decrease in cell viability following cisplatin treatment in islets. And this suggests that the effects that we're seeing on insulin secretion following cisplatin exposure aren't necessarily being driven by overtly cytotoxic effects, but rather through disruptions of the machinery and the mechanics involved in hormone secretion itself. And so, this profound decrease in oxygen consumption that we're seeing in cisplatin exposed islets suggests that the whole islet itself is being impacted by cisplatin, which in turn could likely be affecting all of the endocrine cells and the beta cell specifically, which is influencing this dysregulated insulin secretion that we're seeing. But on that note, too, a lot of our readouts are done in whole islets, and we really need to understand better the effects of cisplatin on other endocrine cells in order to understand how paracrine signaling and the islet as a whole contributes to beta cell specific defects. But one piece of data that was beta cell specific was the electrophysiology, where we saw that cisplatin exposed beta cells did have increased exocytotic capacity under low glucose conditions, which was very odd. And that too could be contributing to the defective insulin secretion that we're seeing. But also it points to the fact that there is for sure at least some beta cell specific effects cisplatin is having.
Very interesting. I think that the fact that cisplatin did not have a toxicity effect is very interesting and also very important, right?
Mm-hmm.
Really really seeing that these drugs can have such a broad effect, right? We're using them for this one little thing, or not one little thing, but to treat, cancer, but we're having widespread physiological effects.
Yeah.
And really building on those changes, you identified some changes in gene expression that were linked to oxidative stress, insulin production, apoptosis. Were there any particular striking or unexpected gene changes that stood out to you as key players in beta cell dysfunction?
Yeah, there were quite a few. We did look at a lot of gene expression and transcriptomics. And I think as someone in islet biology, the first thing that really stood out to me was the downregulation of both the insulin genes and the proprotein convertase genes in mouse islets following cisplatin exposure. I think the downregulation of those genes really goes to show what profound effects cisplatin is having on beta cell function and insulin production, in turn affecting the insulin secretion as well, most likely. In fact, the transcriptomic analysis that we did demonstrated that hormone secretion, and specifically insulin secretion itself, was the most enriched pathway and had a numerous differentially expressed genes that were downregulated within cisplatin-exposed mouse islets. But on another side of things, we also see that there's a decrease in beta cell identity genes such as MAFE and urocortin 3 and NKX 6.1. In fact, we see a downregulation of MAFE and urocortin 3 in both mouse islets as well as human isolated islets as well. And this downregulation of beta cell identity genes could suggest that there's potentially beta cell dedifferentiation happening. And based on that, we also see that a loss of beta cell identity has been documented in senescent beta cells as well. And with the increase that we're seeing in BCL2L1 and CDKN1A, which are two transcripts that play a key role in the senescence pathway of BCLXL and P21 respectively, taking all of this together, it could also suggest that cisplatin is promoting a senetic phenotype rather than an apoptotic phenotype, which could potentially explain why we're not seeing an increase in cell death following cisplatin exposure, but we are seeing such profound effects on islet function itself.
Very nice. Dr. Bruin, are there any other results that you would like to highlight or tell us about where you want to take this next?
Yeah, I would say, I think Lahari's done a really good job of summarizing the kind of the key findings. Maybe I will just emphasize, how important it is that the effects that we're seeing in mouse islets are translating to human islets. So far, we've only been able to test 3 donors. Well, at least at the time of the paper, we had tested 3 female donors. But we're currently expanding that because we think it's really important with work in human islets to be looking at a broad range of biological variability, across many, many donors. So we're in the process of working on that now. In terms of, next steps, we definitely want to keep working on cisplatin to understand exactly how cisplatin is disrupting insulin secretion. There's still lots of questions on cisplatin. But we're also interested in understanding how other classes of chemotherapeutics might be impacting islets as well. And I would say the other big gap that we're hoping to fill is in the clinical side. And this is a different direction for my lab. We don't typically do clinical work, but we have recognized that we really are lacking the clinical data to show that patients who are undergoing chemotherapy have disrupted beta cell function. So there's really good, compelling data looking at the link between chemotherapy and metabolic syndrome, but most of those studies don't really look at insulin or, measures of beta cell function. So to the best of our ability, because this is also difficult to assess clinically, we are in discussion right now with an oncologist and an endocrinologist for how we could design a clinical cohort to get at this question in patients. And so hopefully we'll be able to move that forward in the next year or so, but that's kind of the stage we're at. We're discussing and planning and applying for funding to do that clinical work.
So it is the ideal situation. You start with an observation in preclinical work, and then you actually get to take it to the clinical aspect and answer the same questions.
Yeah, exactly. And I will say, I mean, Lily is the one leading this side of the project, and she's presented, most of the data from this publication to the oncologist and the endocrinologist. And I mean, they were very easily convinced that this is something we need to translate into the patient population. So we didn't have a difficult time convincing them that, this is worth doing clinically.
That's great. Well, Dr. Jennifer Bruin and Lahari Basu, we want to thank you for joining us today to discuss your exciting work on cisplatin exposure and insulin secretion. I encourage the listeners to read the full article in the April issue of Diabetes. Thank you.
Today, we're going to discuss April's paper of the month, titled An Alternatively Translated Isoform of PPAR Gamma Proposes AF1 Domain Inhibition as an Insulin Sensitization Target. We're joined by the corresponding author, Dr. Amit Majithia. Dr. Majithia is an associate professor in the Department of Medicine at UCSD. Welcome, Dr. Majithia. Congratulations on your paper, and thank you for being a part of this month's podcast.
Thanks for having me, Kirk.
So, Amit, tell me, we're throwing a question in here that we didn't give you in advance. It's an icebreaker question. So I'm going to put you on the spot. Tell me who you find to be the most inspirational scientist, live or dead, and why.
Oh, wow. That's a toughie because there are a lot of good ones. I think one of the most inspirational scientists who really affected how I think about genetics and diabetes is Steve O'Reilly. And as you guys may know, he's been knighted for his work.
Yeah, that's a good one.
As one of the first people to discover insulin resistance genes in humans. And I've had the chance to meet with him, to work with him. And so I find his work to be very inspirational.
That's a good one.
That's a great answer. So Amit, before we jump into the article, we like to give our authors the opportunity to highlight how science is really a team sport, right? So is there anybody in your group or any collaborations that you want to acknowledge before we get going on this?
Absolutely. So I'm the corresponding author, but this was a bit of team science that spanned well over a decade, because the initial observations that led to us thinking about this and then working on it in my lab came from when I was a postdoc. And so my own mentor, David Altshuler, supported this work. He's not on this paper because it came a long time afterwards, but then Jason Flanick, who did participate in this work, he and I were postdocs together at the Broad Institute before I started my lab at UCSD, and we continue to collaborate today. And then I also want to acknowledge Sven Hines, who's a colleague in the Division of Endocrinology here, who's really an expert with transcription and really helped us learn a piece of technology that we were able to use to prove some of these thoughts that we had about this novel isoform.
That's great. So now, can you give us an overview of your research program and then how this specific project kind of grew out of it?
Yeah, so my lab is focused on translational aspects of insulin resistance, right? So what does that really mean? It means that we want to understand the molecular, the genetic basis of insulin resistance in such a way that we can actually apply it to people and improve their lives, right? So insulin resistance is very hard to measure. As people know, to truly measure it, you have to be brought into the clinic, get insulin infusions, 2 IVs. It's complicated. And so we use surrogate measures, but it's the thing that happens before most people get diabetes. And even if you don't get diabetes, it increases your risk for fatty liver, for atherosclerosis, for cardiovascular disease. So we think it's really important. And so our lab is focused on finding genes that may cause insulin resistance or actually protect people from insulin resistance that we could potentially develop into therapies. And then novel ways to use genetic data, genomic data, wearable data to get at, do people have insulin resistance without having to come into the clinic and have this expensive and laborious test.
That's really cool. So this work is focused on PPAR gamma. And so we're going to jump right into your paper. So for those of us who may not think about this nuclear transcription factor every day, can you introduce some of the really critical, basic structural domains that this protein has and that you were interested in this particular paper?
Yes. And, to give a real introduction of PPAR gamma would take more than this whole podcast because You guys may know it's an
Sure.
incredibly famous gene. It was, discovered in the 90s in Bruce Spiegelman's lab by Peter Tontenose. And this gene is a nuclear hormone receptor. It is a central controller of fat cell development. So if you take it out of a mouse or a man, fat cells don't develop. And if you put it into most kinds of cells, they will kind of turn into fat cells. And so that's functionally, but then the protein domain itself, the protein structure itself has several domains in which it shares with other so-called PPARs and nuclear hormone receptors, which it's nuclear, so it can get into the nucleus. It has that kind of signal. It has a DNA binding domain that binds certain sequences in the DNA. It has transcriptional activating function, so it turns on genes and recruits co-activators, so it has those domains. And then it also has a ligand binding domain. And so one of the only drugs that actually impacts insulin resistance directly, thiazidolidinones like rosiglitazone, pioglitazone, that we don't use as often anymore, but were major anti-diabetic medications, work through this gene. They actually bind and activate PPAR gamma, but were discovered before PPAR gamma was discovered. And then the last piece I should mention is this AF1 domain, because that's a major subject of our paper. In that domain, it was thought that perhaps it had something to do with binding to other helper proteins, co-activators, co-repressors, but that part is loosely structured and had actually never been crystallized. So there's a published crystal structure of PPAR gamma, but they actually had to take that part out because it was so loosely folded that it wouldn't let the protein crystallize. And so we only know the structure of the stuff after the AF1 domain.
Yeah, that's a great introduction. So as I read this paper, one of the things that I really liked, one of the novel, cool things was the use of CRISPR-Cas in this THP cell line, right? And you induced, I guess induce is the right word, variants, one cell at a time, right? To evaluate each nucleotide of the gene. Is that correct?
Yeah, that's correct. The original experiments that we did in the THP1 where we essentially used DNA synthesis to print out every 10,000 copies of PPAR gamma. Each one of them had a different amino acid, one different amino acid, okay? And then we introduced those into THP1 cells, and if the cell got an active copy of PPAR gamma, it would be sorted into one bucket by flow cytometry, and then if not, it went into another bucket. And that's how we very rapidly characterized what every amino acid change would do in PPAR gamma. So the purpose of that project, which I did when I was a postdoc, was to help create a lookup table for if people had PPAR gamma mutations, would they get insulin resistance and lipodystrophy, or was their mutation benign? And then what happened with this project was that in the garbage file, essentially, when we did the data analysis, there were all of these PPAR gamma genes that through errors in synthesis, they had one extra letter, which would create frameshift mutations and stop them. And so by looking at this essentially thrown away data, we saw that if you got early mutations in PPAR gamma, there was still high function in THP1 cells. But after you get those mutations in the DNA binding domain about 1/5 of the way in, then it's as expected. If you break the sequence, then it doesn't work. And so that's what kind of inspired this work. And then around that time, CRISPR-Cas was invented. And so we were wondering, is this an artifact of us like printing out genes? So would this actually work if you edited the actual genome of a cell? And that's what we did in my lab, which is do that systematically using CRISPR-Cas over all the different exons of that gene, and found essentially corroborated what we found from printing out the synthetic copies of the gene.
And then you went back and confirmed this in adipocytes as well, right?
That's correct.
Yeah, so giant amount of work. I mean, this is really, really impressive.
Thank you. And, I think the original version of the paper didn't have adipocytes in it, but the reviewers rightly said, you want to talk about insulin resistance and PPAR gamma, why is all the work just only done in macrophages? We want to see it's relevant to adipocytes, and they were right, and we did it, and it panned out. So,
right. For the record, I was not reviewer 3.
All right. So you essentially introduced 95, to just highlight again the amount of work, 95 different variants into these THP monocytes, correct?
That's right. The original number was 10,000, and then the CRISPR-Cas was 95.
Okay, I see. All right. So then how did you tell us a little bit more about how you characterized the PPAR gamma activity in these studies?
There were a number of different assays that we used. And so in the original 95 that we did, we used a more high throughput assay, which is we put one variant into one cell, and then so each variant would be represented in thousands of cells. And then we sorted the cells according to whether they contained evidence of an active version of PPAR gamma or not. So the cells get sorted into two buckets, and then by sequencing, we figure out what variants actually landed up in those buckets. And so you can kind of reconstruct an activity map, right? So we started there, and that's what clued us into that there might be an alternative translational start site. And so then we honed in on a few variants that really exemplified that activity, the early stop codon variants and the later stop codon variants. And then we studied those in a variety of different ways, looking at PPAR gamma as a transcription factor, so PPAR gamma mediated gene expression, and then in adipocytes, PPAR gamma is a master regulator of adipocyte differentiation. So we looked at differentiation. TZDs, which activate PPAR gamma, are known to affect cellular insulin response. And so then we looked at insulin stimulation in these cells and how well they initiated the cascade downstream with AKT phosphorylation. So a number of different assays.
Right. And so it was, I guess, help me understand. It's really, you normally expect to see these severe decreases in like CD36, the target gene of PPAR gamma, right? But in these specific variants, you had unexpected activation of that target gene, suggesting that you had activity of PPAR gamma still, right? And that variant now you've named it M135, is that correct?
That's right. So what happens is if you introduce a stop codon in a gene and you only get part of the gene, it usually doesn't work, right? And what we're finding is something odd, that if you do that later in the protein, that's exactly what happens. Like you don't get any PPAR gamma activity. But when we were doing that early, you were getting PPAR gamma activity. And the reason for that is that at methionine 135, which is encoded by an ATG, the cell is able to rescue the expression of PPAR gamma by producing a shortened copy of the gene that bypasses that stop codon or frameshift mutation that's happening early. And so it produces this shortened protein that has the DNA binding domain, that has the ligand binding domain, that has the ability to activate genes, but is just missing that AF1 domain.
Yeah, that's really cool. And what a great example of not giving up on a project when you see something that you don't expect.
I appreciate you saying that. Yeah, I don't know if it was it was really like an academic curiosity that we just felt like we had to figure out. And yes, it did ultimately take over 10 years to sort it out.
Very nice. Let's see. Are there any other thoughts or points that you want to leave us with, as any other findings you want to highlight?
I think one thing that we didn't get a chance to fully explore, but I thought was very interesting, is that long before we did any of this work, there were these studies where they knocked out PPAR gamma in mice. And if you knock out 2 copies of PPAR gamma in mice, they just aren't born because it's required for placental development. But they knocked out 1 copy. And so if you create human mutations in PPAR gamma in 1 copy, you get insulin resistance. But in the mice, in certain types of mice, it seemed that if you knock out 1 copy, the mice were actually more insulin sensitive and they gained less weight. So it didn't match what was happening in the human. And we think that's because the way the mouse was knocked out was they created a gene construct in an early exon, and there was this bypass phenomena. So those mice are now gone. But if we had a chance to look at the tissues of those mice, I would predict that they would have M135 expressed in them. And so it kind of put together like this little mystery in the field that we found.
Yeah, very cool.
Yeah.
Well, Amit, we want to thank you for joining us today, for sharing your expertise, and for discussing your recent work on this novel PPAR gamma isoform. I encourage the listeners to read the full article in April's issue of Diabetes.
Welcome to another edition of Sweet Talk. I'm Kevin Williams, and today's episode is dedicated to the life and legacy of Dr. David Wasserman. Dr. Wasserman was a pioneer in metabolic research and had a lasting impact on our understanding of exercise physiology, glucose metabolism, and diabetes. From leading Vanderbilt's Mouse Metabolic Phenotyping Center, or the MMPC, to setting the gold standard for metabolic research, Dave's contributions continue to shape the field today. Joining us today to celebrate his legacy are some of the people that knew him best. We have Maren Laughlin from the NIH, who was involved with the MMPC since inception. We have Julio Ayala, director of the Vanderbilt Mouse Metabolic Phenotyping Center. And we have Shawn Burgess from UT Southwestern Medical Center in Dallas, Texas. And of course, I'm also joined by our co-hosts, Kirk Habegger and Darlene Sandoval. Drs. Laughlin, Ayala, and Burgess, thank you for joining us today.
Thank you for having us.
Thank you. It's such an honor.
You know, what really stands out to me about Dave's career is his breadth of work and his lasting influence. He had well over 200 publications, many of these appearing in American Diabetes Association journals. I counted at least 40 publications in the Journal of Diabetes. His impact also extended to consensus statements that were handed out by the ADA. There was a 2006 consensus statement on physical activity and type 2 diabetes, and an earlier one in 2004, both of these being cited well over 1,000 times. These contributions help shape how we study and understand diabetes, exercise, and metabolism. And we're going to discuss more about the science in that. But before we get into that, let's take a moment to reflect on Dave as a person. I know he was known as a sharp mind. He had high standards, and he had the ability to make people laugh. And so as an icebreaker, we wanted to kick this off. I would love for each of you to share something about Dave that stood out to you, maybe some favorite memory, a story that captures who he was, maybe a moment that surprised you. Let's start off with Julio.
I don't think I can pinpoint any single moment that stands out because there's so many with David. I think one of my fondest memories of David, and this is kind of generic, but spending time in his office was always a treat. Because there were two certainties whenever you would go into David's office. One is that you would be there for far longer than you thought you were going to be. And the second is that you would probably end up talking mostly about something that had nothing to do with the original reason why you stopped by his office. And I use that as an example because it really highlights the personality that David had. David was very knowledgeable in a lot of different areas, and he could talk about a lot of different things. And this is why you would spend so much time in his office. And it wasn't just about science. It was about politics. It was about sports. He loved sports, particularly baseball. He had a very sharp and almost photographic memory. He could tell you, for example, the starting lineup of the Chicago Cubs in 1962. Actually, he could tell it to you from probably their inception. It was always a treat to spend time with him in his office. And I think that really highlights, the fact that he was a very sharp scientific mind, but he was much more than that. And the other reason why I bring up the fondness that I had in spending time in his office is really something about David, and that's that he actually spent time talking to people. One of the things immediately after his passing that I kept hearing from colleagues all over was how much time he dedicated to them. Spending time on the phone or via Zoom meetings, talking about science, talking about life. He really gave a lot of his time to helping his colleagues and his friends. And I think that's sort of a microcosm of spending time in his office, is the fact that he was so willing to give of his time. And that's really probably one of my fondest memories of David.
Yeah, and I'd really echo that. I got to know him more in the latter part of his life, probably in the last 5 to 7 years. And I reached out to him on numerous occasions, and he was very willing to do these virtual meetings and discuss science. And it was a really great experience for me as well. So he was very giving and a great mentor to not only people, there at Vanderbilt, but outside, some lowly soul like me. So I really appreciated it. Maren, what about you? What maybe memory or story would you have of Dave?
Again, there are so many, and he was such a unique person, a little crazy, a little nutty, a great scientist. I mean, a true physiologist, in the sense that he kind of understood so many different biological systems and so many ways of interacting of different types of information, and so creative with all of that. He was just a remarkable scientist. I remember I'd wander into his office every once in a while, and basically you'd see him sitting slouched down in his office chair with some kind of old hat like pushed down over his face, and he'd kind of push up that hat a little bit, lean back, and give you this impish smile, and just launch into whatever craziness was in his mind at the time. But he never failed to say something really nice and generous about the people in his lab, about the colleagues that he worked with, and about his friends. And after he died, I talked to a lot of people, and virtually, 90% of them said he was my brother. He was my mentor. He was my best friend. And that's the thing that is most memorable about him. I have one other story I wanted to share, which was that the first time I ever saw Goofy David lock in on a scientific problem, it actually just shocked me, because the first time you see an incredible mind kind of at work on a problem is just an amazing experience. And that was David. He could think so deeply and so creatively and in such a focused way about a scientific problem. It was just glorious to behold.
That's great.
I'll echo that, because we call that Scary Dave moments, when he would actually focus and just really go into science, and we're all just waiting for the shoe to drop. It's like, okay, when's Goofy Dave coming out? But yeah, when he was focused, and it was a sight to behold, definitely.
Nice. And Shawn, what about you?
Yeah, it's hard to listen to stories from Julio and Maren and not have an emotional response to all that, because you just, everything they said is just so familiar. But I remember early on when I first started going to the ADA meetings, and the first time you're at an ADA meeting, it's overwhelming. It seems like chaos. You don't know where to go. You're surrounded by all these wonderful scientists, and you're just kind of overstimulated. And I'm walking through the corridor, and I see David Wasserman sitting in a chair, and I go up to him, and in all this chaos, he's just sitting there reading For Whom the Bell Tolls. He's reading Hemingway, and I'm overwhelmed, and I he invites me to sit there, and we talk, and he tells me about why he likes Hemingway and why he likes to read historical novels, because it helps him put things in perspective, and that's how he understands history best. And so you might meet him one day and he's reading Hemingway, and the next day you meet him and he might want to talk to you about glucose disposal. The next day he might want to talk to you about the 1954 San Francisco Giants, or the next day he might tell you a dirty joke. And so he's all of these things. And for those who were fortunate enough to know him well, you would have that entire conversation at one time in random orders, and probably multiple times during the conversation. He was just this beautiful, chaotic mind, and I miss him.
Those were some really wonderful memories there. So I appreciate that from all of you. At this point, we kind of want to jump into some of David's work, right? Shawn, I'm gonna stay with you here. David's research significantly advanced our understandings of glucose uptake and insulin resistance, and specifically in skeletal muscle. Can you take a moment and maybe highlight some of his key scientific contributions in your mind and explain their lasting impact on diabetes research?
I mean, that's a tough one. So I would invite, everybody else to chime in because there's so much to say here. But I think the theme of what David worked on, well, his theme was everything, as Maren said. He touched on everything, but he was really interested in the integrated control of glucose metabolism, the push from the liver and the pull from the muscle, and how these things were coordinated. And speaking of Hemingway, you know, he wrote this very famous review article called 4 grams of glucose. You know, I mean, if that's not your iceberg theory, you know, put into one, I mean, that's just out of this chaotic mind that could go everywhere. You get 4 words, 4 grams of glucose, and it means so much. And so I would, you know, encourage everybody to read that review paper, even if you're not a scientist. You know, he puts things simply, and so it was easy to understand. And he talks about insulin, of course, and insulin stimulated glucose uptake, but he also talks about insulin independent mechanisms and how counter-regulatory mechanisms play a role in maintaining this exact 4 grams of glucose, and how the liver is regulating gluconeogenesis and glycogenolysis to push back against the uptake of muscle. And towards the end of his work, you know, I mean, what he really wants to talk about in that paper is that glucose regulation is not just about glucose transport, that it's about how the glucose gets to the muscle, how the hormones get to the muscle, the role of the extracellular matrix, the role of the endothelia. I mean, he was thinking about these things in 4 dimensions. And that paper is 15 years old, and yet it's still so relevant and still so new and so fresh. And those figures, I mean, from this chaotic mind, this is what Julio and Maren were talking about. The guy who just got done telling you a dirty joke, he wrote 4 grams of glucose. It's beautiful.
I have a question now for Julio, but before I get to that question, inquiring minds want to know, our listeners want to know.
I know where you're going, and I am not going to answer that.
It's Dave Wasserman, editor-in-chief of Cell Onion.
Can I chime in? I heard that it was Kevin Williams. That's
Yeah.
Yeah.
The secret is out.
Yep.
All right, I'll let you off the hook on answering that if you'd like. One of the other things that's really cool about Dave is he's kind of scientific royalty, right? Carleton Wasserman was his dad, and in the 60s he had conceptualized those gears models, right, that really described how gas exchange from the respiratory, cardiovascular, and muscular systems was really important for regulating or for maintaining exercise capacity. And it's Dave took off on that model, right? Can you talk a little bit maybe about Carleton's model and how that might have influenced Dave?
Yeah, David's father, Carleton, was a big influence on David, I think implicitly and explicitly. You know, even though Dave may not have recognized it sometimes, at least outwardly, I think his father really was a huge influence on him. And in conversations that I had with David, I could tell that his father was a role model for him in how he wanted to grow up and develop as a scientist. And so I think it was very fitting that David took that concept of multi-step regulatory processes for gas exchange and applied them to what David ultimately became interested in, and that is, you know, regulation of tissue glucose uptake, that it's not just this one rate limiting step, that it's really distributed amongst different compartments whose importance may change depending on physiological versus pathophysiological states. And so I think the gears representing, you know, transport for muscle glucose uptake, transport, uptake, and phosphorylation really became the center point for David's research in understanding this fundamental process. But at the same time, I think it was kind of tipping his hat to the legacy of the work that his father, Carleton Wasserman, did. So I think, those gears will stand the test of time in many ways.
Yeah. Shawn, do you see, since you're our omics guy, do you see any opportunities, or do you think the metabolic flux field was influenced by that model?
Well, I think so. I mean, the whole idea of distributed control, I think, comes from the flux ideology, and the fact that David was able to embrace that and extend it into something that is not obvious as flux. I mean, because glucose transport, we just think of as kind of the very first thing, and then what happens after glucose is transported, that's what people tend to think of as flux. So he was tuned in to that entire idea of metabolic control theory, and that's what it really is. And again, demonstrates his ability to cross disciplines and do it almost accidentally. It just seems so natural.
And I have a question for Maren. You were around with the inception of Vanderbilt's MMPC. They founded that Vanderbilt MMPC over 20 years ago. And in founding this center, he didn't just build a core facility, he really transformed how the entire field approaches metabolic phenotyping. You have that unique perspective on the founding and the development of the MMPC over the years. I was wondering if you could share a little bit of the early stages of the MMPC, how that initial vision came to pass, and how it's progressed over the years in your eyes.
Yeah, absolutely. And it's kind of amazing. That was 25 years ago now, and I just want to mention that both Shawn and Julio were part of the MMPC at the beginning back then. You were both graduate students, I think, weren't you?
I was a postdoctoral fellow.
You were a postdoc, and
Yeah, I was just finishing my postdoc, or I think somewhere in the postdoc area.
And at the time, the reason that we put the MMPCs on the ground from NIDDK was because we had a lot of people at that point who had adopted the use of transgenic mice, of course, for studying metabolism in diabetes and obesity. And it was becoming clear that these animals, in order to really make them the important central model for studying diabetes and obesity, they also had to be physiologic models. We had to find a way to study the whole body metabolism. Dave was a dog guy. He was trained in large animal models. He could, put a catheter in anywhere, study balance across any organ. He was a real physiologist who had been trained in metabolism using this incredible dog model. And when he came in, he really was not a mouse person, but he had decided that that was the future, and he decided he was going to make it the future. So he basically took the studies that he had learned to do in the dog model, and he worked to translate them into the mouse, but without giving up any of the sophistication that you could have in the dog model. So that was his goal, and that was uniquely his goal. He really wanted to transform the field in such a way to be able to take advantage of the physiology and the ability to manipulate the genome and to do metabolism all at the same time. And so one of the things he decided to do was really to interrogate the best ways of doing these sophisticated exams in the mouse models. He really engaged all of us in this process. He basically pushed everybody to do their best to really figure out the best way of doing these exams, and then to basically make sure that as many people in the world as possible knew how to do it. And if not, if they couldn't learn, if he couldn't teach them, he was going to do it for them. As he really, really cared about the quality of the research in this field. And so he transformed it.
That I feel like that is a big part of his legacy, right? Not just that he figured out the gold standards for assessing glucose homeostasis in mice, but that he wanted to make sure everybody knew about it, right? By publishing articles and spending time on that education side of things. I've always thought that was a really cool part of his work.
I was going to say he really transformed the MMPC into what I consider to be an altruistic organization. And honestly, I hadn't seen that before in the scientific world. You know, people work together well and all of that, but he really took it to a new level.
Yeah, to that point, one of the phrases that sticks in my mind, how David described the MMPC, was that he really wanted the MMPC to be an extension of our users' labs. He wanted the users to feel as if they were part of the work that was going on in the MMPC, as almost as if they had a bench right here in the MMPC. Because of that, you know, the importance of rigor and reproducibility was there, but he also wanted the core, the MMPC, to be more than just a core that would do the experiments and generate data and throw it over the wall to the users and say, here, good luck. You know, the users were part of the experience, and that's why he really pushed for the MMPC to spend the time with the users in the experimental design and the interpretation of the results, so that we felt ownership to what we were doing as much as the users had ownership in the project. And that's what I think has made the MMPC really unique.
Yeah, Julio, how appropriate that you actually just chimed in with that. You've taken over Dave's role as the director of the Vanderbilt MMPC, and I'll say, you know, you have helped us personally in constructing the techniques and interpreting our results, and so that obviously those words of wisdom have hit home and still exist today. Beyond that, do you want to add to the legacy that David left in maybe your specific role as the director?
Yeah, it's shoes that I know that I cannot fill. I mean, David's shoes, you cannot replace somebody like David. But you know, spending a lot of time with him, I've absorbed a lot of that ethos that he had and that I just described. That in his memory, and to continue the work that has gone on in the Vanderbilt MMPC for the last 25 years, our goal is to continue to provide that excellent, rigorous level of service to our users, that they are integrated in the process, and also that we continue listening to our and meeting the needs of the scientific community. That was another important component of David's vision for the MMPC, was to listen and innovate and develop tools that will be useful not just now but in the future. And you know, in my experience with the MMPC, one of the things that has helped us grow and develop is really listening to our users and taking their input, taking their advice. So many of the things that we've implemented at the Vanderbilt MMPC were because of input that we received from our users that we then went on to implement. And so I think that's my vision for the future, is maintaining that rigor, maintaining that personal relationship that we have with our users, listening to them, and then innovating in different areas, whether it's looking at complications like chronic kidney disease or developing tools to interrogate how the central nervous system plays an important role in metabolism. These are things that we are looking to do to take the MMPC to the next step.
And I guess as a quick follow-up, can you envision any new or emerging technologies that you think Dave would just love to bring into the MMPC or integrate into what you're already doing?
Towards the end of his time in the MMPC, we really made a significant effort to establish methodologies using stable isotopes, because you know that's going to be really an important leap forward in our ability to assess metabolic fluxes. He and I had a lot of discussions, particularly when I was recruited and we talked about me being the next director of the MMPC. As I mentioned, he was really excited about the ability to integrate methods for measuring and modulating neural systems to investigate the effects on that the central nervous system and neurons have on metabolism. Those were two key areas that he really was interested in looking at. But then more holistically, I think one of the themes that we've talked about that was important to Dave was integrative physiology, is how all these things are integrated. And are there ways, are there methods that we can develop to look at integrated physiology and not just reductionist physiology?
The next question is for Maren. And as we discussed in the intro, Dave co-authored some highly influential ADA consensus statements on physical activity and diabetes management. So do you think did he have a broader impact on some of the way that NIH looks at physical activity and diabetes management? What are your thoughts on NIH's incorporation of exercise currently or maybe in the future?
Well, I can tell you for absolute sure that he had a huge impact on the way that I think about, incorporating exercise into particularly metabolic studies. For our clinical studies, exercise and lifestyle is always part and parcel of everything that we do. But also, Dave was really instrumental in bringing exercise into the basic science world. Basically making it possible to study exercise at the same time, this integrative physiology concept, studying animal function in that way with the context of metabolic flux and metabolic control. One of the ways that really influenced me was at the beginning, basically I worked very hard to put MotorPack on the ground. And MotorPack is basically a large study to try to characterize human physical function from a molecular point of view. And it was a lot of the conversations that I personally had with Dave about this whole concept of organism function as a part of understanding regulatory control of metabolism that made me think that it was really important to kind of establish the dictionary or the encyclopedia, I guess, of all of the molecules that participate or arise as a function of exercise in the human being in order to inform future studies in that area. We have, in the metabolic world anyway, for decades, we always studied people lying flat on their back, not moving a muscle, to be in, some kind of steady state in order to really interpret the metabolic experiments. But that leaves out, probably 95% of the regulatory control of all of the metabolic pathways. So I think he was instrumental in pushing us into an era where we can really much better understand metabolic regulation, because the ability to add in knowledge about exercise and what's going on metabolically during and after exercise.
Yeah.
And Shawn, Dave's research really opened several avenues of investigation in metabolism and diabetes. What do you think are any current or future scientific directions that are particularly exciting and build directly on Dave's legacy? Are there any emerging areas of metabolism research that you think Dave's contributions uniquely prepared the field to explore?
Yeah, and only Dave could do this. I mean, because as Maren mentioned earlier, he really established the foundations in the beginning in large animal models. And so only somebody who had studied organ-specific metabolism in a large animal model through AV differences, through radio tracers, all of this foundational work really led to our ability to look at genetic models in smaller animals like mice. And in particular, his work in exercise physiology, especially early on, I think established a foundation that for many of us now is just part of our common knowledge. And you hope that doesn't get lost. Some of these things that we take for granted, like the role of glucagon in exercise, or the role of other counter-regulatory mechanisms in exercise, how exercise can stimulate non-insulin dependent utilization of glucose, how exercise can stimulate glucose storage after exercise through the storage of glycogen. And these are all things that I think are becoming more and more important, or at least more and more focused on in the literature. And so I hope that those of us who are working in exercise and how it affects various organs and diseases will remember to look back at this early literature that David in part helped to form.
We also want to inform our listeners there will be a symposium in memory of Dr. David Wasserman at Vanderbilt University on June 13th, 2025, so just in a couple months from now. Julio, can you tell us more about the symposium?
Yes, happy to. Yes, we are hosting a symposium in honor of David on June 13th. That is a Friday at the Vanderbilt Student Life Center. We will be sending out a RedCap link for people to RSVP if they want to attend. We have an incredible lineup of guest speakers, two of whom are here in the podcast today. We have Shawn Burgess and Darlene Sandoval invited to speak, as well as two former trainees of David's, Curtis Huey, who's currently at University of Minnesota, and Lee Kang, who's at the University of Dundee. And this is really a celebration of David, the impact that he's had on science and just science in general. It should be a great event. So please attend if you are able to.
And do y'all know yet, is this going to be in person only, or are you going to have a virtual component as well?
This is going to be in person, but there will also be a virtual component. Yeah.
Nice. And I did note it down, the RedCap. So if anybody is interested in registering for this, the RSVP is www.redcap.link/DH Wasserman. That's D as in David, H as in hotel, Wasserman. Or you can email mmpc@vanderbilt.edu. Thank you to our listeners for tuning in to this episode of Sweet Talk. As always, please like, follow, and subscribe for more diabetes bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.